Good morning, madam. Welcome.
Thank you very much. Thank you, sir, for inviting me in this lecture. It will be great. I heard a lot about
So,
ISF Moga. This is the first
So,
opportunity.
welcome, madam. So,
so
very nice to meet you. I think you are quite comfortable in the Raebareli.
Yeah,
And so, madam Sobni ji, she is the heading of the NIPER.
yeah, ma'am. Dr. Shubhi Saraf is the director of our NIPER.
Yeah, she is my senior in Sagar.
Okay, right.
I did my B Farm M Farm from Sagar.
Okay, very nice. She is very, yeah, like intellectual wise also, and as a human being also, she is very nice. Both. Yeah,
Actually.
very nice.
Yes, Dr. Rangraji.
yes, sir. So, shall we start, sir?
Okay. So
Okay.
shall we start like formally because you are going to YouTube and record it and later on it will be online.
Yes, sir.
So
Yes, yes,
initially
sir.
actually when I said yes, so I don't know it will be go to online. So it will be recorded.
Okay.
So it's fine, right? So
So, okay.
yes, please.
So, okay. So, good morning to all of you. Myself, Dr. Naresh Rangra. I welcome you all to this ISFCP sponsored dialogue series. So, before starting the session, I would like to request Dr. GD Gupta sir to say some inauguration remarks, sir. Start the session.
So thank you very much, Dr. Nareshji. So first of all, I welcome today's expert. She is very dynamic and dedicated toward the profession. So first of all, I welcome Dr. Savaji at ISF College of Pharmacy from core of my heart. And I am thankful to you for accepting our invitation and you have given your precious time for this interactive session. As I know that in those days, most of the professors, they all are the busy in their respective schedule because day to day, not only now, the responsibility of the teacher to day classes as well as the practicals, what we all are in a very small speedy motion. So that is the number of the things is the required to the going in the academic system also administrative as well as the academic daily routine activity, publication, so many things. So thank you very much, Dr. Savaji. And now, as you know that ISF College of Pharmacy regularly doing the something special for the professional. One of them, this is the our ISF dialogue series under the IQAC, and Dr. Siddharth Manji is the taking care of this series. And I am very happy to announce that I want to extend this IQAC series up to the 365 talk on this platform so that people they will take the benefit. The purpose behind of this one, because I know that the person have the very less time, so these types of the lecture I want to the especially and justify and very short form in the you can say recorded so that we can put on our YouTube channel and faculty or student they will take the benefit. So I hope that you can understand the purpose of this ISF dialogue series. The purpose behind only this one, those person who have some of the special capability, we want to the connect the peoples and we can connect them to provide this information which will be the useful to each and every professional. So without taking time, I again welcome you, madam, and thankful to you for accepting our invitation. I am thankful to our coordinator, Dr. Rajesh Rangraji, for provide the opportunity to interact you on this platform. Thank you very much, madam. And
Thank you.
over to Dr. Nareshji.
Thank you, sir. Thank you for this inauguration remarks. So, let me introduce to you today's speaker, Dr. Saba Naqvi. She is working as assistant professor at Department of Pharmacology and Toxicology or Regulatory Toxicology. She obtained her master's degree in toxicology from Jamia Hamdard, New Delhi, and further did her PhD in 2013 from Jamia Hamdard in collaboration with AIMS, New Delhi. She has done her postdoctorate from Jamia Millia Islamia and worked as DST women scientist at IIT Roorkee and has published more than 37 research articles and book chapters in various reputed international journals like Elsewhere, Springer, Ville, Frontiers. She has filed 2 Indian patents, and she has guided 35 master thesis research work, and currently 4 PhDs are enrolled under her. She is the member of many national and international societies like Society of Toxicology, MRSI, Indian Academy of Neurosciences, and Indian Science Congress Association. Dr. Saba has honored with prestigious Women Scientist Award from DST, AMR DXC, Summer School Winner Award sponsored by DBT, India, and University of Edinburgh, UK. Currently, she is working in the field of neuropharmacology, nanomedicine, nanotoxicology, and synthesized ceramic calcium phosphate nanoparticles for suicide gene therapy in cancer therapeutics. She is working in the area of toxicology following OECD guidelines in the area of acute and subacute chronic toxicity assays, reproductive toxicology, xenotoxicity, immunotoxicity, and neurotoxicity. Her lab is also working in nanotheranostics for the induced toxicities. The environmental toxins are thought to be the major contributors in the various diseases like cancer, neurological and metabolic disorders, etc. So her lab is explored diverse research tools including in vitro and in vivo animal models of neurodegenerative diseases using nanotheranostics approaches including animal imaging to study the molecular mechanisms of disease and to screen novel compounds for treatment. So, with this, I welcome Dr. Saba Naqvi on his talk, Regulatory Guidelines of Animal Research, with special emphasis on the Committee for the Control and Supervision of Experiments on Animals. So,
Thank you.
welcome, welcome.
Thank you very much for inviting me. First of all, I would like to extend my gratitude towards your director, Gupta sir, as well as you and your team who has given me this opportunity to online platform. It will be, as sir told, yeah, it will be very easy to come online. Anytime you can spare 1 hour and half an hour to come, although it is very difficult earlier to go somewhere and not right now also because we are busy in other responsibilities also. So it is a nice, I hope that I covered the topic very well, as well as because this is related with the guideline. So I would like to say maybe the few information is missing because every day the guideline is there and something is updated everywhere. But I will focus basically on the Indian guideline which we are following, the regulatory guideline for the animal research in this lecture, that is the CCSEA. As I would like to tell you one more thing, that recently it is the earlier it was the CPCSEA. Now the purpose has been removed from the website and this is CCSEA. So today, first of all, I would like to welcome all on the behalf of all NIPER Raebareli fraternity. So, as I told you, we are going to discuss about the regulatory guideline for animal research. Not with all the guideline, we are focusing on the animal research. So, the question first has been arise what is why the animal is, because these days a question has been raised by various agencies, by various regulatory bodies, that why the animal is required. So earlier it was right now also it is a thought like the significance of animal research in scientific advancement. As we know, most of the animal that we are using, the rat and mice, primate, non-primate, that is in fact the mice and humans share more than 98% of DNA similarity. This is one of the major reasons why we have started animal research. So nothing so far has been discovered that can be substitute for the complex function of living, breathing, whole organ system and pulmonary circulatory structure. So rival, although the animal is a very complex system, but still we are not able to find out the because sometimes the human trials are not able to be successful based on the data on the animal. But animal are still, because we are taking animal models, because there are many diseases that people are suffering, that is the health issues like the cancer, diabetes, and other heart diseases, other neurological disorder, Alzheimer. This rat and mice and this animal system mimic all the organs, so that it will be very easy to find out the physiological function. Similarly, that this animal is a short lifespan. Due to the short lifespan, we can assess, or in the many generation also, not in the single generation, the other generation also, we can check either this, what is the effect of any metabolic effect, any genetic effect. So this is the animal, like animal has a short lifespan. This is also one of the reason. So why we are testing the drug, why the safety and efficacy of the drug are used to do on the animal model. But as you know very well, like this is the ethical concern is very very much high, and these days there is a question arising all over the world, even in India also. I will come to that point also. Like, is it appropriate to use animal for research purpose? Yes. And various like drug research, and as well as the cancer research, various biology toxicity testing of any xenobiotics, as well as the educational purposes for students also. We are using different rat, mice, guinea pig, rabbit. So the human animal right activists always raise a question that use of animal just because they are we consider that they have a lower moral status, so that we there's always a fight between the animal right activist, as well as those who are using the animal for their safety and efficacy purpose. So they are saying that the animal should be treated with the same respect as people. No one should be able to force them to perform any task, to kill them only for these purposes. So, but still, animal right deploy the same argument. Many advocate are having this kind of like arguments. They that human beings does not give us sufficient ground declare animal are being morally less significant. So why they are using the animals? These are the ethical questions, ethical concerns which are arising by the several agencies, by the governing bodies. So that's why our animal regulations are highly required to justify what the work you are going to be done. Is it justified or not? Or you are going to kill the animal just for the sake, because they cannot say anything. So that's why the ethical committees are there all over the world. And India also, there's a ethical committee, IAEC committee, that I will come in this lecture. So ethics in animal experiments, it must be very very important. Like that, ethics is very much important in everything, not in the animal research, but we are talking particularly in this section, that is the use of animal, so for an experimentation. So a clear scientific purpose, if you know that your purpose is not solved without using the animal, then only you can go ahead with the animal research. If it is possible to it will be clear, or it will be solved, your purpose will be solved without using that animal, so you can go ahead with the without animal. So least amount of pain and suffering, because of animal cannot say anything. You cannot do the things like this, that you can give any kind of pain or suffering, any injection. So we have to adopt a method that should be very least amount of pain. As well as the human treatment of animals, human treatment is very much important. Not only the treatment, they are just because animal, we have to give the proper care, and it must be trained by the it must be supervised by the trained personnel. So significance of research ethics that ensure proper treatment of animal experiment to avoid undue suffering of animal, that is very important for the ethical consideration. All over the world, the legislation around the animal research is based on the idea of moral acceptability of the proposed. So morality is very important here before using any animal. That's why the regulation has come, ethics has come. You cannot use the animal just without any reason, or just because you want to check. At present, most ethical guidelines are dealing and they are considering that the animal experimentation is justified because of the significant potential benefit to the human being. So what we are thinking that the human should be benefited, so that only in that case we will use the animal for this research purpose. These guidelines are often permissive of animal experimentation regardless of the damage to the animal, as long as human benefit are achieved. So these are the few points. Now, um, okay, so ethical principle, even we follow these principle, but still there are few changes are going on continuously. Step very small step India is also taken recently by the new drug and clinical trial rule 2023. There is an amendment made by them, by the government of India. He passed amendment where they aim to stopping the use of animal in research, especially in drug testing. The amendment authorizes researchers to, instead of non-human, they focus on the non-human or and human relevant method, that is can be organoid, that is like a 3D model, that is organ on chip, and as well as the in silico techniques. Even all over the world, not in the India, even other country at the European Union, in 2021 they have taken a step towards the resolution which also promote the 3R. As well as the US passed one more act in December 2022. US passed the FDA Modernization Act 2.0. The FDA Modernization Act, that is the 2.0, make a game changing, maybe a game changing legislation, so enabling drug registration without the absolute requirement of use of animal in safety toxicology assessment. So it is very much important that the safety toxicology assessment can be done by the other mean, by using the 3D organ printing, organoid, lab on a chip, or organ on a chip, as well as the other advanced computational in silico technique. It can be, it is accepted now, not by India, by the other countries, as well as the other act. So like in 2022, South Korea introduced a bill called the Vitalization of Development, Dissemination, and Use of Alternative to Animal Testing Method. So there is a now alternative to animal testing is widely looking by the various governing and regulatory bodies, that if we can use it, it will be good. But if we do not have any choice, then we will go by the animal usage in the experimental research. In Canada also, they have also amended its Environmental Protection Act to replace, reduce, and refine the use of animal for the toxicity testing. So these are the concept of 3R. Actually, we are continuously telling we should use the 3R, 3R, 3R for what? For the reduce the pain of the, reduce the suffering of the animal. The aim is that. So these are the like the refinement, as well as the alternative to animal. What is the alternative to animal testing? Like what Indian government has recently on in March has suggested in the amendment in the clinical trial rule, that you can use the computational subject, computational studies. You can use the in silico, as well as the ex vivo or in vitro. So these kind of things that you can use it, as well as the refinement is very important. What is refinement? For example, to use the other better handling, so that the process that we are adopting for the testing of chemical on the on the rat, it should be refined. It should be like, if it is avoided by using the injection, you can avoid, you can give the oral. If it is avoid, what is the refinement? Refine your skill, so that the minimum suffering and pain will be there, as well as the reduction of animal. If you have, like for example, for any study, you are using 6 animal or 5 animal per group, but if it is possible, you can control and reduce the animal by 3 as per the rule. So you have a valid reason that you want to reduce the animal, and the 3 will give the result, statistically good result, and you can reduce the animal. This is the reduction. So these are the various method. um, in vitro digestion, organ in chip, these are the various method, that is the invertebrate in silico model. let's come to the main regulatory body that is overseeing ensuring the ethical treatment of animal in research in India. We are focusing. We are just a minute. Okay. We are in this particular talk. We are focusing on the animal research on regulatory body, which is governing the regulatory body in India. This is the Committee for the Control and Supervision Experiment on Animals. This is a committee of Department of Animal Husbandry and Dairying. Sometime you don't know even which ministry it comes, so it must be know it should be coming with the Ministry of Fisheries, Animal Husbandry. So if you will go through this lecture, you have a basic idea who what is the CCA body. CCSEA body. So that it is, as I already told you, this body is has been made by the experts to that to reduce to ensure that animal are not subjected to unnecessary pains and suffering before, during, after the before, as well as during, as well as if possible after the performance of experiment. So this has been formed by the committee formulated the breeding of experiment on animal control and supervision 1998. This is the amended in 2001 2006 and regulate the experiment on the animal. So what is the aim of guideline? The aim of this guideline, which has been prepared by the expert, is to ensure human and ethical treatment of animal. This must be it. It reflects that our Indian governing body, regulatory body, also very much concerned about the usage of animal. You cannot use animal like any animal in one group, like 8 animal, 10 animal, 5 6 animal, whatever the animal you want to choose, and nobody is there to regulate you. There is a body which is the Indian government has assigned a body that will have a eye on you, that you are using animal, and ensure that the human and ethical treatment of animals should be there while facilitating legitimate scientific research involving experiment on animal. The purpose is similarly the same as it is formed on the basis of the international norm regarding the usage of animal, by updating the regulations and streamline simplifying the procedure while ensuring ethical use of animal, reducing infliction of pain, stress on animal during experimentation. So these are the objective that is given on the website of the IAC, CCSCA. The IAC is the Institutional Animal Ethical Committee that is formed by any institution who has having this animal facility, and that is registered animal facility. So if you want to carry out any animal research, you first of all you must have this institutional animal ethical committee, where a few members are there, that ensures that experiment shall be performed in every case by or under the supervision of a person duly qualified. So the qualification is also very much important in this committee. Here, the degree or diploma holder in veterinary sciences, medicine, laboratory animal science of university or institution organized by government for the purpose. So a veterinarian is very much important in this committee, that scientists from this inside the institute as well as the scientists from outside the institute, one chairperson is there, one member secretary is there. So there's a many members are there, like one veterinarian is there, full-time or part-time veterinarian as per the requirement of the institute or the facility, as well as the scientists from the inside the internal IIC committee are there, as well as the outside internal, that is the nominated by the CCSEA SEA few members are there which must have a look on the internal IC committee that the whatever they are proposing it is ethical or not, or they are doing with the due care and humanity or not. Your institute is there. So that's why we have also a few nominee, two or three, one main nominee, one link nominee. These nominees are there which have a look on the animal care. So as far as possible experiment, the animal should be given like what kind of anesthesia should be there. Is it is sufficient power to prevent the animal feeling pain. So this is all required to know any uh, before starting any animal experiment. So animal should which is in the course of experiment under the influence of anesthesia should not be uh, uh, they recover very seriously, involving serious suffering, ordinarily destroyed while still. So experiment on animal are avoided wherever it is possible. So it is already I told you the animal experiment should be, I think, okay. So experiment to animal are avoiding wherever it is possible. It means like if it is not much required and it can be done with the help of in silico or some other in vitro tools, you can avoid it. As well, the guideline also permit now. So these are the functions of CCSEA. Registration of establishment conducting animal experimentation. First of all, if you want to do the animal experiment in your premises or in your institution, in your academic institution or wherever you want, in for example in some industry, first of all you have to register. They register your establishment with the CCSEA animal experimentation or breeding. If you want to do breeding, that is a different one. But if you want to carry out the experiment in your premises, in your academic institution, or in industry, whatever the establishment is there, so registration is very important. After that, the approval of animal health facility on the basis of report of inspection conducted by CCSEA. So few members are nominated or few chosen. They will come and inspect your facility. Either your facility is appropriate or not for carrying out the animal research. It is ethically as well as the what are the norms are there. They are your facility is following or not. After that inspection, they allow for the approval of animal health facility. Then, as I told you, selection and assignment of nominees for the institutional animal ethic committee for the registered establishment. So registration, then approval, then the assignment of the nominees from the CCSEA. Permission for them, they will give the permission for conducting experiment involving the use of animal with the help of the internal IIC committee member. So IIC meeting will be conducted regularly, and there the other the forms be or they will propose the inspection will be there. And then only after the inspection, after the going through the form B, they are allowed to have the experiment. Action against establishment in case of, this is also the function of CCSA. If you are doing any illegal, any legal norm stipulation violate, if you are violating or illegal something, so the action will be there and your establishment also loses this registration. Conduct of training program for the nominees of the, this is also the function of CCA, as well as the conduct and support conferences. These are the function of CCA. In short, I will go in detail also, but this is the flow of CCSEA. So that ethical review and approval that I told you. So a researcher must obtain the ethical clearance from the IAC committee before conducting any experiment. This is very important. Institutional animal ethics committee should be there. So those who, any PI, any principal investigator, any researcher, those who want to do work in the animal, they must have go through the IAC committee before starting or before conducting any animal experiment. Detailed research proposal must be submitted to IIC for review and approval. Then the alternative, if there is any alternative to animal testing, this is encouraged as well as suggested. So that in vitro method or computational simulation, if it is there, this is more highly encouraged. Experimental animal, what kind of animal, what is the choice of a species, what is the age as well as gender was, as well as the how many animals. This should be very much clear in the form B. So number, as age, weight, sex, purpose, summary, all should be there. Animal should be of a species that is appropriate for the research question. So this is also important, like the research question. What is your research question? Accordingly you will design your study, accordingly you will use the species. Housing and care, and anesthesia, pain management, that I will come in the later slide. And euthanasia is also very important. You can have to reduce the pain and distress to animal. Reporting and record keeping is very important time, important point, because anytime when the animal house inspection facility inspection inspector has come, that is usually a main nominee of IAC committee, that is the main nominee assigned to the assigned to your IC committee. That should be, they are used to check the record of animal usage, procedures, everything. They used to check whatever, either you are doing the things rightly or not, what is the outcome, risk form D, what the method and result of accurately and transparently. So record keeping is also, archiving is very important part of this committee. The training, the, it must be all the person, all the researcher which is involved here should must have properly trained. So animal caring and animal handling care, ethical consideration, they should know properly, then only they should know what is exactly they have to do. So oversight, inspection, collaboration, information sharing, so these are the things that is the part of the process of IAC committee. These are the current CCSEA committee members. Joint secretary. This is only for the. This can be changes. Can be changing as per the new rule. So, this committee currently working. So, the goal of these guidelines to promote, as I told you, with the human care of animal in biomedical and behavior research and testing, with basic objective of providing specification will enhance animal well-being, as well as the quality, as well as the more knowledge about the for the human trial, because this is a preclinical studies. Preclinical study, the aim of the preclinical study to provide the data for the clinical studies, so that this is the rationale of choosing the animal. There are few important forms are there, that is form A. Form A is for the registration, as well as for the, if you want to register your establishment, you have to fill the fill the form A. That form A is available here. You have give the detail of the establishment, either it is a government, private, what is the name, where the, what is the address, and all the detail you have to fill it and submit to the CCA website online. This is very mandatory. Without that, your form will not be considered. So I told you veterinary care. Veterinary care is very much important. That's why, as per the guideline of Indian Regulation Animal Research (CCSEA), they have clearly said, like, adequate animal care must be provided is the responsibility of a veterinarian. So you can see here. Okay. Educate daily observation of animals. This is the role of the veterinarian. They should do the daily observation, check the animal health, their behavior, their well-being, so that if any you can say any mishappening is occurring, they should report promptly, as well as the proper care should be given. So monitoring, healthcare and husbandry, reviewing the protocol, all should be taken care of by veterinarian. That's why veterinarian is highly required for this. This is the form B. Form B is, as I told you, form A is for the registering, as well as the for breeding purpose of your establishment. But if when the form A is form A is failed and you are registered, your normal IAC committee is formed, then now if you want to test any chemical or anything or any drug, whatever the xenobiotic, so the actually you want to do any research, you can say, so you have to fill the form B. Like here you have to give the detail of the name and address of establishment, registration number that is already assigned to you, as well as the name and the address of the breeder from which the, if you have the facility of your own, you are going to utilize your own, or so you are going to utilize from some other nearby institute, where the animal will be kept, where the experiment will be performed, what is the date and duration, very minute details they have asked to make it clear that what you are going to do is rationally right or wrong. So they want to check only. Then if they are satisfied, yes, you are going to do here, where you are going to do, what kind of study you are going to do, who is going to do, what is the title, who is the principal investigator, what is the experience of the person who is going to do the, like they have some experience in handling the animal or not, or they are just submitting their protocols. So the protocol should be checked very thoroughly, where you have to given the experience also, those who are going to involve in these studies, as well as the animal required species, strain, age, weight, gender, rationale for the animal usage. If you are going to do any surgical procedure, you have to written what doses, what site, what animal, what volume of the doses you are going to do. So the purpose of these form is only not to restrict the people, researcher to do the research. The purpose of this is only to have do the research in a justified way, so that the undue pain should be avoided. This is the form B. You can even go, and they will finally they will give the invigilator declaration is there that you are using this for research proposal is submitted, uh, and the certificate is given by the chairman, as well as the member secretary and the main nominee. The chairman and the member secretary is generally the institute only, and the main nominee of CPC SEA, that is now the CCSEA, it's signed, and after that certificate you can start, you can do your work. So animal procurement, that is also a very important point. So I told you, like animal can be taken from some other institute if you are not having the breeding license. So the animal must be acquired very lawfully. As per the CCSEA guideline, the animal health surveillance program for screening incoming animal must be carried out to assess animal quality. Method of transportation is very important. So you can use the AC vehicle, and with the proper space, as well as the height and space of the material used in the transport box, that should be taken care while procuring the animal. So when the animal has come, what you have to, as per I'm all telling whatever I have taken from the CCA guideline. So this is very important, and when you are taking out the animal, the quarantinization, stabilization of the animal, and the suppression of animal, because you all know very well, like when corona happened, what happened, we all go through the quarantine process. In the same way, the animal also go the quarantine is a separation of newly received animal from those already in the facility until the health status of the newly received animal have been determined. So, for example, an animal has come from some other facility and they are infected, and you don't know, and they are mixed up in the, like the infection, in the case of corona. That's why what happened, you have to separate the person for 7 days, 14 days. In the same manner, you have to separate the animal in for a small and lab animal, it is 1 week to 1 month, whereas the large animal, large animal like the cat, dog, monkey. So the 6 week, for example, for the large animal, it is of quarantine period is 6 week, as well as the regardless of duration quarantine, you have also check the veterinarian should also check the physiological, psychological, and the stabilization before their use. They are psychologically fit or not, they what the type of feed they are taking, they should be same or not. So physical separation may also cause many problem. So hence before starting these experiment, you have to check all these points, the quarantine, stabilization, as well as the, okay. Mm, guess I have changed something. Slide. I have stopped the slide somewhere. Give me 1 second. I guess I have done something.
Ma'am, the sharing wording for this slide is upside arrow.
Hello.
Yes, ma'am.
Now,
Can you?
now it is visible.
Still not, ma'am. Slides are not visible.
I think now it is visible. Is it visible, sir?
Still not, ma'am.
Just ये रुक गया देखिए जरा.
Is it visible now? Okay, it's visible.
इसको छोटा कर दीजिए. बहुत बड़ा हो जाता है इसकी वजह से दिखती. मुझे तो वही बैठना है. Okay, sorry for disturb. Maybe there is a technical glitch. So now it is audible as well as visible, right?
Yes, please continue.
Okay, sorry for that. So quarantine stabilization and separation is one of the things, as well as the surveillance, diagnosis, and treatment and control of disease. If the animal have some which has come from some other institute or in your institute only, or in your organization establishment, you can say any word here. So it must be observed for the sign of illness, injury, and abnormal behavior by the animal house staff very carefully and critically. If the animal has died, the post-mortem examination and sign of illness as well as is also reported very minutely and reported promptly, so that it will not spread to other animal also. And if animal have some contagious disease, it must be isolated from the other animals, so that the spread of the disease should be restricted to other animals. Animal care and technical personnel, the personal hygiene is also very important here, because those who are handling the animal, the people trained in laboratory animal science or to provide both formal on job training must be hired, so ensure the effective implementation of the program, so that the animal care staff maintain a high standard of personal cleanliness is very important, so that you have to take care of animal like your child. So you cannot like this you go and handle. So personal protective equipment, shower caps, change of uniform, footwear should be there. The use of disposable gloves, mask, head cover, coat cover, shoe covers. This should be there, so that, and uh, it is used whenever you enter into the facility. Personal should change the clothing before entering the animal house facility, as well as the garment or the lab coat you are using worn in the animal room should not be worn outside the animal, as well as the other person should are not allowed to use the cosmetics, cream and perfume in the animal rooms. So these comes under the ethics. So whenever your student are going inside the animal like this, they are using perfume, they are not tying your hair, so you should say like this is as per the rule, you should not do this. Animal experimentation involving sometimes some hazardous agent also. For this, institutional biosafety committees are there. That committee review the use of this biohazardous agent, if it is of like can be used or not in your institute, or this kind of thing is good for human as well as the animal, or for the rational use of this hazardous agent, they can allow. But in the along with the biosafety committee, you have to take the permission with the institutional animal ethical committee too. So this is the also like the like in IAEC, IBSC is also the institutional biosafety committee, where the head of the institution or his nominee, three or more scientists. In the same way, in IAC, the chairman will be the head of the institution as well as the HOD or nominee. Three or more scientists engage in DNA work or molecular biology, a member with medical qualification. But here is a one difference between the IAC and the IBSC. In IAC, no medicals are not mandatory, but in IBSC, a member with medical qualification is a mandatory. So one member nominated by the DBT. This is also not the same way. The member is in biosafety committee. The member is from DBT, but in this IAC committee, the member is from CCSEA. The physical strain is also like that. To avoid, we have to check the sometime the animal with the collection of sample or other clinical experimental manipulation, we use the restrainer. But the prolonged restraint of any animal, including the chairing of non-human primate, should be avoided unless essential to research objective. So you cannot use the different strain for only for the sake of using a strain. You have to be very use of minimum period required for the restraining in the restrainer, because period of restraint should be minimum required to accomplish the research objective. Animal should be placed in a restraint device, should be given training to adapt to the equipment. You cannot do like this, you can enter the animal and bus first time and then you can do the thing. No, you have to give the proper training, like a person or a child can give a training. In the same way, for the animal also need some training. Provision should be made for observation animal at appropriate interval. The presence of any lesions, illness, or severe behavior change should be dealt with temporary or permanent removal of animal from the strain. So if you have feel like some lesions or illness or some behavior changes, sometimes they are very violent, you have to do it very fast. So facility is also as per the guideline, the building material. I don't know how much time is left, so that I will restrict my uh, this lecture. So building, yes.
So you have sufficient time, ma'am. You can.
Okay, right. So, what is the physical facilities? The physical facility is also very important. That is the infrastructure for any. The if you want to do the conduct the animal this animal experimentation, you must have the building material which is made of durable, moisture proof, fire extinguisher, pest resistant. This is the building material. As well as the corridor, it is not very narrow. It is wide enough to facilitate movement of personnel and equipment. Animal room door, proper fit, rust free. These are the animal room door. So everything you can see that very much clear by the CCSEA. If you want to open the facility, not to with the CCSEA, in all over the world, the facilities, the government agency which are looking for the animal uses, they have their norms and regulation if you want to do the experiment. So you can see the floor, even the floor is also mention what kind of floor should be there in the animal house: smooth, moisture proof, and a skid proof floor, so that drains proper drainage is very important. No sloped floor is there. The storage area, there's very much important, the design of feed and bedding, how what kind of storage areas are there. Sometimes what happen in animal facility, if you are not doing the properly store the animal, the fungus will be there, and that aflatoxin or fungus will be developed in the feed, and that will ultimately alter your the result also, because the health status of the animal is altered because of the this feed and these conditions, environmental conditions, and then ultimately the result is hampering. Environment, what kind of environment? Like for example, you know very well the animals are very sensitive, so you cannot give, like sometime it is in India, especially you can say like it's sometimes it's raining, sometimes it's too cold, sometimes it's too hot, but for the conduction of the animal experimentation, the animal is must be housed in the temperature and humidity that is the 18 to 29 degree. The humidity should be maintained 30 to 70%, as well as the ventilation, everything you can say very minutely they have given the guideline. And when the inspection occurred by the when the this CCA person nominee will come and check your animal house. They will check all these point. Is these point are fulfilled by the facility or not? If it is fulfilled, then only they will give you the license to carry out the work. They will give you the registration. So power and lighting, safe installation, fluorescent light with emergent backup facility, noise control. It is like the noise-free environment is highly required. If your institution, your industry, your establishment is nearby to some place where the lot of noise are there, because what happens if the noise are there, you have to make the concrete wall are more preferable, because in that case the animal will be disturbed, their behavior will be disturbed. So housing is one of the things that provide space that is adequate, permit freedom of movement of normal postural adjustment. Like we as a human being required some space. For example, we required one table, one chair, one in your institute, one lab, one space, one resting place. So in the same way, one house, one room. In the same way, the animal which are living inside the animal house facility, because they are restricted to the cages. So the cages should be like this. They must have the sufficient space so that they can move around on in the animal cages. So provide a comfortable environment. The comfortable environment should be there. Provide a escape proof enclosures that confines animal safety, provide easy access to food and water. This is very important point, that the access to food and water is not there, they cannot survive. So we have to take care all these small point, but this is very important. So provide adequate ventilation, meet the biological need of animal, maintenance of body temperature, urination, defecation, reproduction, keep the animal dry and clean, consistent with the species requirement, as well as the facilitate research while making good health of animals. Activity, what kind of activity? If you are caring some specialized locomotor pattern to express their natural habitat. If you are using monkey, for example, so what they are doing, animal are held for long period. If you are taking cat and dog for your experimentation, so that you have to give the proper space, like artificial trees, rope, bar, perches are appropriate for non-human primate, as well as the cages are often used to short confinement housing of dog, so that I so isolation of sick dog and metabolic study, so that you have to maintain this proper environment, healthy environment, so that they can behave normally, and then only they are able to give the right result. Because if the animal is not is in a stress condition, they are not able to work to give the right condition, right results. So food is also very important. As you know, the food is how much important. If you are getting the right food, you are happy, and no, you are not having any kind of disease. But if you are not getting the right food, what happens? The same way behave animal like the as human behavior. We are already using this term many times in this lecture and all the documents you have seen. Human behavior, like for example, they must be given a good diet, a palatable diet that can be approached, that can be eaten very easily. It must be non-contaminated, as I told you. It is not having the any kind of contamination. It must be nutritionally adequate. It must be, that means just say you can see like for example, you need everything, all the vitamin, you take the fruits, you take the milk, you take everything to make the nutritionally fit. In the same way, the diet which you are giving to the animal must be nutritionally fit, adequate, devoid of contamination of urine and feces. That the bedding is very important. That will become the it should be devoid of that. That's why the always the animal feeding will be kept upside. That cannot be having contaminated with the urine and fecal material. Sufficient amount to ensure normal growth in immature animal and to maintain normal body weight, reproduction, lactation in adult. So one of the simple thing that you are in toxicity studies also, what we are checking the weight of the animal. So body weight is one of the indication that the animal is fine or not, is health is okay or not. So you can see free from chemical and microbial contaminant. Animal feed should contain moisture, crude fiber, crude protein, essential mineral, crude fat, carbohydrate, so that appropriate nutrition. So what is diet? Like for example, in our case, what we need. In our case, what happened, we are telling the term adulteration. All the diet what we are taking should be free of adulteration. In the same way, the diet should also be free given to the animal with the heavy metals, lead, arsenic, cadmium, nickel, so that or any other toxin or contaminant. Exposure to extreme to relative humidity, unsanitary condition, light, oxygen, as in the deterioration of feed, so that the proper consumption as well as the storage of feed is very much important. Bedding, so you know very well, if the bed is not good, the you are not able to sleep properly. Next morning you are not able to behave properly. It's a very simple human behavior. As I told you in the our first slide also, like the animal are very much biologically, physiologically similar to the animal, to the human being. In the same way, if the bedding of the animal is not good, it is like wet, or it has some contaminant, or you can say fungal and fungus and anything. So what happened, they are not giving the right results. So it should be non-toxic, sterile, easily stored, and stable, non-detritus to animal and cane, non-injurious. Sometimes it must be like that, so that cause injuries also. Bedding should be very good, non-nutritious. ऐसा भी ना हो, like for that, for example, they can take the this bedding material as a food. It must be not nutritious, not palatable. So water. Just you, as I already told you, like for example, bedding is very important, food is very important, food is very important. So what about the water? You know, many diseases are just because of the water. In the same way, animal also behave similarly. If you are not giving the proper water, have continuous access to what fresh water. Well, if you have a not, if you not drink the full two or four bottles of water, sometimes you have some health conditions to develop. In the same way, the fresh, potable, uncontaminated drinking water is also required, highly required for the animal. Be provided. It must be accessible to them also easily. With water free from microbial contamination, watering devices such as drinking nozzles, automatic water. These are the devices that we are used routinely to ensure their proper operation. So water, you can see very clearly that how water, bedding, and the physical conditions are important. So I told you one more thing initially, that the record keeping is very important. Why? In any case, when you are doing the some facilities are also GLP certified. That is, the GLP certification means you can do the experiment and give the result to the industry or whatever the institution they are asking for the GLP study, so that it will go further with this result. So GLP a very important point is the record keeping. So what are the record keeping are there? You must be from, not even in the GLP, even you have the your animal house not registered with the GLP, you must have the good record keeping. That must having the animal house plan, which include typical floor plan, all fixtures, animal house staff record, both technical and non-technical. It must be very much important that whatever the staff are involved in your animal house must having the good health. They are not suffering from any disease. Health record of staff and animal. SOP is one of the most important thing you must have in your animal house, and it is checked by the inspectors also. So these are the relevant to experiment, care, breeding and management of animals, breeding, stock, purchase and sales record, minutes of the IAC. Minutes, what is minute? Minute is like whatever you have done, you have approved some protocol, the protocol must be signed, that protocol is signed, it must be signed by all the member, the minutes of the IC meeting must be there, so that it and it should be uploaded time to time whenever the IC meeting was conducted to the website of a CCSEA. Record of experiment with the number of when you have done the experiment. What is the record of experiment? That is the form D. We have already gone through the form A, form B and then the form D. Form D is like the record of animal acquired and experiment performed. For example, you have cleared the form B for 30 animals, 60 animals, 50 animals, whatever. But what when the date of entry of this animal to your institute establishment, what is the number of animal required? For example, you have taken the Vistar rat, you have taken the SD rat, this is the species, you have taken male, female, this is sex, this, for example, you have taken 2 to 3 months old, young, or you can say of old. So it depends on the age, whatever the age you have to mention ki what date of entry, what is the number of animals, 50 animal, for example, name and address of registration of the breeder from where you have acquired, as well as the date and particular of grant of permission or either committee. So that this is the form D which give all the detail that when you have started, from where you have taken, when it will enter, when who has performed and who has done the, who is the PI of that authorizing. So this is a very important part of CCSEA form D also. Mortality and the post-mortem record should be there. If any untoward actions are there, it should be maintained by the facility. Training record of staff involved in animal activities, water, feed and bedding material, analysis report. So these reports should be there. Rehabilitation record. So, as I told you, the SOPs, institute should maintain the SOP ascribing method with regard to the animal husbandry, maintenance, breeding, animal house activity. Generally, it is SOP in the SOP can be of any type, but generally it is of name of the author, title, date of approval. For in this case, I'm telling you, SOP is the standard operating procedure of any instrument. It can be of an instrument also. It can be of any animal house facility also. So you have to do the SOPs with that. I'm not going in detail. Anesthesia. This is very important point. So anesthesia should be that anesthesia should be given to the animal that causes the minimum pain, right? It can be of intramuscular, IV, intraperitoneal. You can see different mouse, rat, hamster, guinea pig, rabbit, cat, dog, ketamine, phenobarbital, thiopentone, urethane. So these are the anesthesia. Euthanasia, the painless killing that should be given. So breeding and genetics, there's one another form, that is the breeding and genetics, that is the form C. Record for the breeding acquired. This is the initiating a colony or breeding store must be procured from CCSEA registered breeder. It must be the genetic makeup and health status of animal is known. So for this, the form C will be there. I have already told you a lot about the this IAC, that is the Institutional Animal Ethic Committee, which is registered by the CCSEA and which is constituted and operated in accordance with the procedures specified for the purpose by the committee. So these are the member which I told you, like the biosafety committee. Here is the IAC committee members: one biological scientist, two scientists from different biological discipline, one veterinarian, one scientist in charge of animal facility, one scientist from outside the institution, Once a non-scientific socially, yes, here you can see in IDSC, socially aware nominee was not there, but here you can see the scientific, non-scientific socially aware person is there. One or two representative from the main nominee or link nominee from the CCSEA. So this is the same thing. The experiment not performed merely for the purpose of acquiring manual skill. This is very important. For only for the sake of manual skill, you cannot perform. They must have a proper justification. Must have some clear vision why you are going to do. So these are the initiation, CC approval process, ethical clearance, IAC review, and then the ethical approval, then the CCA submission. This is a normal process which I told you. You develop a research. When you are going to do any research, you have to do the prepare the form B, that is a research proposal. This proposal, this form B should be committed for the ethical review to the IAC committee. The IAC committee will review the and ensure its scientific and ethical validity, as well as the justification of the animal uses, experiment design, ethical consideration. Ethical approval should be given on the basis of the what proposal you have given. If they are satisfied by the justification you have proposed, if modification is required, they also sometimes you have given like you will use 8 animal per group, but sometimes they are saying no, it is not required, you can reduce the number of animal, it should be like 5 animal per group or 4 animal, or you can use the number of same number of animal in the control. So these various kind of modification as per the suggestion, or sometimes they propose like you can do it later, first generate some in silico data, some any kind of suggestion that is suitable for the proposal, they are given to the by the committee members. Then the CCA evaluation approval decision, like the approve the proposal if it meet all ethical and the research execution. If it is approved, the research education is there. Like for example, the researcher can proceed with conducting approval. Ongoing compliance throughout the research, researcher must adhere to the. It doesn't mean that you have got the approval. It means you can do anything. During the while performing your experiment, you have to throughout your research, researcher must adhere to CCSEA guideline ethical consideration. Regular reporting and record keeping are very much essential and the final approval. So now we have already described in detail of what our country, that India is looking for the animal research. But in other country also we have a very like other bodies are there which are having eye on the those who are using the animal for research proposal. Like I'm discussing only few of them here. This is majorly what USA, like or European countries. So United States has enacted two national laws that impact laboratory animal research: the Animal Welfare Act. Actually, they are using the Animal Welfare Act and the Health Research Extension Act. So Animal Welfare Act enacted in 1996 and amended in 1970, and continuously few amendments are coming, as I have told you in the earlier slide. So these are few information which is given to you. The Animal Welfare Act is the primary federal law, basically, there which regulate the treatment of animal use in the research, as well as the administered and enforced by the Animal and Plant Inspection Service under the US Department of Agriculture. Here also we are using the Animal Husbandry and Dairying Ministry of Fisheries. In the same way, they are having some ministry which is handling this, administrating this laws, set a standard for the human care. The same thing we are showing and we are observing, that the human care and treatment of animal in research, exhibition, transportation. They have the PHS policy, that is a public health service population policy, required compliance with the guide and care of use of laboratory, widely accepted reference. As well as in the same way, we have the IAEC, they have the Institutional Animal Care and Use Committee, a slight different. So animal institutional animal ethical committee we have, they have the IACUC, that is the Animal Care and Use Committee. Therefore also reviewing, approving, overseeing all the research going on the animal in the institutions, and compliance with the regulation and animal, as well as the 3R principle. So these are the National Institute of Health. They are also looking for the oversees the welfare of the animal in the US. Now the EU, European Union regulation of animal research. So they are also having the same kind of body, animal research in the European Union. They also directive on the directive of on the protection of animal use for scientific purpose. The final aim is also to directive to replace. Now I've seen yesterday only the site of European Union. I have taken this point also from there only, that to replace all animal research with the non-animal method. They have taken all the they have also taken this initiative to replace the uses of animal with the non-animal method, that is the organoid or through computer simulation. In the same way, the replacement, that is method to prevent or help replace the use of animal, reduction and refinement. They also follow the 3 R's. Even all the regulatory bodies, they follow the 3 R's. Now again, in cosmetic also, in 2013 the European Union regulation wide ban on animal testing for using for the cosmetic, for complete ban since 2013. They also follow the same thing like our Indian society or Indian organization follow the species, what the species they can take, like mice, rat, zebrafish, frog, rabbit, cat, dog, and non-human primate. These are the species you can take it for your research purposes. What is the procedure? Procedure should be like this, so that even you can in species. I would like to add one more thing, that the non-human primate can also be used in the limited number of research purpose, not for the all purpose, but in a limited number, right? So if your purpose is not solved, if the research cannot be done in other species, then only non-human primate can be taken. But the rehoming is also there. So rehoming is like that, if it is not sacrificed fully, then you can rehome this, and rehabilitation is also there. The procedures is the same, like the either invasive or non-invasive is called the procedure that we are going to do. When it lead to the level of pain, suffering, distress, to reduce the procedure is the aim of these bodies. So that animal husbandry and care I have already detailed in our country. In the same way, they are also follow the animal husbandry and care, that food, water, environment, enrichment, care should be there. Their health status should be monitored. Overall well-being is the focus of the committees. In Canada is also the having the same kind of structure, having different name of the bodies might be different in different countries, but the motto, the aim of the all the bodies are the same: to reduce the pain, to give the human care, to follow the 3 R's. What I have observed, might be I'm wrong somewhere, I might be I missed somewhere something, but what I observed throughout the all over the world, not even in India, they have followed the 3 R's, as well as they follow the reduced or painful method, so that the animal can be treated morally. The Canadian Council of Animal Care, a national institution charged with stabilizing and upholding standard of the moral use, as I told you, moral use and care of animal in science, overseas animal research in Canada and the mainstream, as well as the Canadian Council of Animal Care. This is the recognized organization of a standard, maintain standard, recommendation करते हैं, recommendation of the moral use. It makes certain animal research is carried out ethically with a focus on animal care. So ethics are very important here. Either you are using the animal or not using the animal, but you have to follow the ethics. So it also provide rules. The Canadian Council of Animal Care is also providing the rules and regulation for the housing, veterinary care, and experimentation. They also providing for the guideline to and recommendation to the academic institution, animal care committee, researcher, AEC. That is the institution. This animal ethical committees are there. In our this is IAC. In US it is ISUC. Here it is AEC. It also the same way, the same manner it work and guarantee that proposed research complies with the CCAC regulation advancement. In Canada, you can see the same thing. In Japan also, there are some laws out there, which is the Ministry of Environment. They have treated So administrative guidance are there, how to govern the law concerning protection and control of animal. They give the guidance, they give the regulation. They also follow the 3 R's, emphasizing the elevation of pain and distress as well as humane death of animal using these institutional guideline. So Australia and UK, Australian Code of for the care and use of animal for scientific purposes. They are also using serve as the basic legal framework for the animal research restriction in Australia. The code offer recommendation for the moral and humane treatment of animal. They are also more or less similar to this uh, this US guideline. And they're also having the National Health and Medical Research Council. This is the council, well-known Australian government organization, like in our country, in change of fostering and managing the national health and medical research. They are monitoring the promote ethical and animal research for its regulatory monitoring. The regulations are there, guidelines are there in different country, and we have to follow with the ethical use of that. In the same way, China is also having the laboratory animal management regulation. You can see and go to any country, who are using the animal, they must have some guideline. Although the name is different, like here, the laboratory animal management regulation. So that, like, but you can see the same point everywhere. The ethical and even I have taken these point most of the site from site only. So they follow the 3 R's. They have the ethical review and approval. Institutional Animal Care and Use Committee or comparable reviewed organization give their ethical blessing before researcher can use this animal in China or not. Korea is also having the same way, having the Ministry of Agriculture, Food and Rural Affairs is the responsible overseeing and regulating animal research in South Korea. They also ethical review and approve the usage of animal. So in short, you can say, if you want, I have given a very short overview of all these guidelines, but if you want to go in detail, you can go to this site, and USFDA site, where the Animal Welfare Act is there. Animal Welfare Act history and digital collection. You can take the different download the books also in India, CCSEA, because I have taken this one from the site, but it is still CCSEA. So you can download it, the CCSEA compendium, as well as the guide for the European uh, guide for the care and use of laboratory animals. So these are the various books. As apart from this, many books are there. I have mentioned only few which I go through, but you can also go through the other books and links also. So, in short, I would like to say the usage of animal is not bad in my view, but it must be rationally defined. It must be justified, so that we cannot use animal just for the sake of this, like for some skill, or just want to check either the work will be done or not, or for the repetition of the work. You have to, if you want to do, if you want to kill the animal, you have to be very specific. You must be very much clear in your aim. You must give something to the society, not for that they cannot say anything, you can do anything with them. So very animal supporters are there. Although I'm also the animal supporter, but I'm also doing the work on animal research. So few of the work, because we are also doing of my lab, I'm doing the work in vitro as well as the in vivo both. So I'm the supporter of both first, but I would like to use the work on this is the lab of my work. Like in short, I will use to tell like the we are doing the in vitro work, so that I support the this hypothesis that animal also need moral support. But although if you are getting the result, I also used to do non-invasive method for the imaging, as well as the various synthesis and characterization of novel neurotherapeutics. Here, this is our lab. Few of the students are very old, but now the new batch is also has come. With this note, I would like to thank you very much for the organization for inviting me, and maybe I'm quite helpful in giving some uh, informative, in giving some information about the Indian guideline basically. So thank you very much for this, and I hope that this lecture will be useful to the student who have joined me. Thank you very much, everyone. Thank you to the director, ISFCP Moga, as well as the sir who is organizing and the team organizers who is organizing all.
So thank you for this mind-opening session, ma'am. So it was a wonderful session on CCSEA guidelines. So on behalf of IQAC ISFCP cell, I extend a really vote of thanks to today's expert speaker, Dr. Saba Naqvi, ma'am, who spared time from her busiest schedule to grace this event, ma'am. And today we had an opportunity to hear your views on the topic regulatory guidelines of animal research with special emphasis on Committee for the Control and Supervision of Experiment on Animals, that is CCSEA guidelines, and this will surely be going to encourage us in our future research events. So,
Thank you, sir. Thank you very much.
thank you, ma'am.
So, shall we go to any other question is there? So, or either you can email me also, or if any question is there, it is most welcome. And all the details which I have taken, it is not from my side, it is from the website on the CCSEA website. So I guess I tried to give the right exact information. Uh, if anything left from my side, please pardon me for that. Thank you.
Thank you.
Thank you very much.